This video is about the polymerase chain reaction PCR, which has the following major components: master mix, genomic DNA, primers and nuclease-free water. In the first column, the volume is for one reaction mixture. You can multiply it with the required number of the reactions. The total volume for one reaction should be 25 microliter. This is the master mix, and these are the primers I am going to use. Always start your work by sanitizing your workplace. I sanitized mine with 70% ethanol. Align your primers, master mix and nuclease-free water in one rank, and then label your PCR tubes according to your sample numbers. Set out the pipette for the required volume. Take out the master mix from the vial and add it in one appendoff. In this appendoff, we will make the whole mixture. We don't add genomic DNA in this appendoff, as genomic DNA will be added in the PCR tube directly. We will add master mix, nuclease-free water and primers in this appendoff. Now add nuclease-free water in the same appendoff. The technique to add is don't double press the pipette. Always do it by single pressing the pipette. It will increase the efficiency and efficacy of your results. Now add primers one by one. I have 3 primers. I'll start from primer number 1. Then I will add primer number 2 and then primer number 3. Do pipetting by single press. Don't double press the pipette to avoid bubble formation. Now I am adding primer number 2 in the same appendoff containing master mix and nuclease-free water and primer number 1. If you have 2 primers and you don't have 3 primers, then add the volume of primer number 3 in the nuclease-free water volume, as the total volume of the reaction should be 25 microliter. Now I am adding primer number 3. Gently tap the appendoff and then add mixture from the main appendoff in the PCR tubes you labeled initially. 23 microliter of the mixture will be added in 3 PCR tubes one by one. After adding this mixture in the PCR tubes, genomic DNA will be added. 2 microliter of genomic DNA will be added in the respective PCR tubes according to the sample numbers. If the temperature of your lab is hot, then you have to perform this activity on the ice. But if the temperature is cold or normal, it's not needed. The technique is same here. Don't double press the pipette. Always single press the pipette. I have 3 samples, so I will add 3 DNAs in the 3 PCR tubes. Now it's time to put up these tubes in the thermocycler machine. Set out your new protocol and temperatures with time accordingly. I will set temperature for denaturation, annealing and extension, and then I will set the number of reactions which will be performed. You have to set out the time for each reaction as well. The volume is 25 microliter. Save your protocol. After saving your protocol, put up the PCR tubes in the wells, close the machine and run the reaction. Thank you for watching.